it's my pleasure to welcome Erika Raven to give her presentation on the cytoarchitecture and brain-wide connectivity which of insula, and which reveals its topographic.
Thank you so much for the introduction. Okay. I have nothing to disclose. And so, as Nicola mentioned, today I'll be talking about the insula, or the so-called hub of interoception. And so interoception is the perception and integration of signals that describe the physiological state of your body. And so this can range anywhere from maybe hunger pangs that you're feeling as we're approaching lunchtime, all the way from a socio-affective representation of like a pleasant feeling you get from a gentle touch from a friend. So effectively, it's linked to an astonishing number of roles. And as you might imagine, its dysfunction has wide-ranging implications for both physical and mental health disorders. And so if we zoom out to look at the macrostructure of the insula, here looking at both human and macaque side by side, you can see that it's tucked away beneath the frontal, parietal, and temporal lobes, an ideal location for integration within the brain. And now I would like to zoom into the microstructure. So here on the right, I have a flat map representation of the insula from the macaque monkey. And you can see that it's comprised of many different subregions, and we can parcel out these different subregions based on their cytoarchitecture. And this essentially means what is the cellular density and laminar profiles of those cells within the cortex. And the flow of interoceptive information that's coming into the brain can be mapped onto distinct subregions based on their cytoarchitecture and assigned functional roles. And so if we think about information coming in, so it comes in through the granular cortex, and so this is encoded with primary sensory input of interoception and body representation. And as you see from this histological slide, it's very organized, and it importantly has a specific layer of granule cells called layer 4. And then information flows into this granular cortex. So this is a very integrative area, and these regions have been associated with roles such as interpersonal processing and social behaviors. But at this time, the cytoarchitecture becomes less organized, and so layer 4 is more poorly defined. And then finally, in the most anterior and ventral area of the insula, we have the agranular cortex. And so this is kind of the most unusual. It's encoding for interoceptive awareness and visuomotor output. And at this point, there's no layer 4, and it's actually the most thick part of the insula, and also throughout most of the brain. And so this collection of subregions with their unique cytoarchitectural characteristics and functional roles represents a very interesting collective of highly integrated regions throughout the entire brain. And so can we unravel specific networks of the insula? Well, in order to do this, we need to establish a direct link from both the cytoarchitecture to the whole brain. And this is essentially a very technical challenge to merge these disparate scales. So thinking about the microscale, we have highly specific information, but this requires postmortem tissue, and it typically is restricted to either small areas such as subregions or maybe a slice. And then at the macroscale, we have the whole brain. We can take repeated measurements over time. We can even integrate this information with functional information, functional MRI. However, we do not have the resolution of cells, so we lose that specificity. But going to a translatable animal model, in this case the macaque monkey, we can actually seek to bridge across these scales. And so here we use the macaque because it's highly utilized within neuroscientific research, but also has similar characteristics in terms of cytoarchitecture and connectivity to the human brain. So let's go to the workflow. And so here I'm presenting both a data set and a workflow called the micro to macro macaque, MicMac. Sometimes hard for me to say that, which is essentially an acquisition of multi-scale and multi-modality data across the entire brain. And what we did is we aligned all of this data within a three-dimensional space. And so this allows us to go down to the level of cells to do segmentations of subregions, all the way up into a three-dimensional volume to make inferences and downstream analyses. And ultimately, because we work within a central space, we can project our results and inferences to a cohort-level, population-level design. And so for the data itself, so this is a single subject. It's a 10-year-old female rhesus macaque. And so it is multimodal, but today I'll be focusing on diffusion-weighted imaging. And so zooming in on the insula, you can see we have some directionally encoded information that will be important for tracking our pathways. But this data set also includes ex vivo MRI, which is at much higher resolutions. And so now zooming in on the insula, you can see that we have signal even into the cortical gray matter. So this will be important for our application of looking at insula tracks. And then finally, we have microscale information. So this goes down to the level of cells or oriented fibers. And so bringing all of this together will be extremely important for our question. That being said, there's a lot of pre-processing that must occur. So there are geometric distortions, intensity corrections that must be applied across all of these different scales of data, nonlinear anatomical alignment across modalities. For example, we have 2D histological slices coming into 3D volumetric space. And so all of this work is done in close collaboration with Claude Lepage from the MNI. And he has a lot of experience working on the big brain data set, which is a whole brain histological data set in the human brain. And so once we have this model, this single subject model across scale, Joey Charbonneau went through at 2 microns on the Nisselstein data, so that's essentially looking at cytoarchitecture, looking at those individual cells, and divide the 15 unique subregions per side. And so I have on the right side, you can see the different cortical types, so these are the agranular, dysgranular, and granular subregions. And then within the model, we can project those into volumetric or surface space. And each subregion is then used as an origin point to establish this mapping of long-range connections throughout the brain. And so we use a technique called tractography. So this is performed in the subject space using a high diffusion weighted data. And then we, for establishing the probabilistic connections throughout the brain, we have these streamlines and we constrain them by known anatomical boundaries, such as the gray matter-white matter interface. And so this is the result from the ex vivo data set, the MicMac network representation. So on the left, you can see all the subregions represented, but then I parse them out based on cortical type. And right away, you can see that there is overlap between different areas, but also distinct representations. And so going to a different view, we have 3 subregions representing each cortical type, just as an example. And I show sort of photo booth style, going from anterior to posterior aspect of the brain. And here in this coronal plane, you can see that there are distinct representations as you kind of scroll through the images. We can then map the projection endpoints, so looking at the density of where streamlines interface with the cortical surface. And as you have a darker color, so that purple color, that means that there's more streamlines interfacing with the surface at that location. And so here we can definitely start to see an emergence of distinct patterns. So for example, for the agranular subtype, we see more orbitofrontal cortex, some superior temporal gyrus, and then going down to dysgranular, a more posterior aspect of the superior temporal gyrus, and then finally with granular, more somatomotor intersection. And then importantly for the insula, it has a lot of, it loves to talk to itself, so there's a lot of projection endpoints that are represented within the structure itself. And so the next thing we wanted to do is, now that we have this model, is to try to extend the coordinate mapping from the single subject design to a cohort level. And so what we did was we matched individual subjects' MRI data to the cortical surface and redefined a central coordinate system, allowing for translation from this model to additional subjects. And so here are the results from a group of subjects, 16, so 3 males and 16 females, age range of 7 to 20 years with an average range of 14. So just from these are fully adults, so it's basically middle age to older age. And the image acquisition and preprocessing were identical to the data that I showed you previously. And so here, just looking at the projection endpoints as a binarized mask with this color encoding that I've been using, you can see patterns starting to emerge. And so this mapping to us essentially occupies distinct areas based on the subregion that is represented by, kind of suggesting a topography similar to other primary sensory regions. And we did a comparison to track tracing study. So this is a pair of studies by Meslin and Mufson from 1982. And these were extremely intensive studies where they had track tracing injection sites at the anterior, middle, and posterior aspect of the insula. And it's important to note that track tracing data actually encodes directionality. So I've shown the afferent and efferent projections. And this is in comparison to tractography, where we actually don't have that. So tractography is bidirectional. But when you look at the correspondence between track tracing and this bidirectional tractography endpoint representation, we have good agreement. So the main observations here is that we did observe a topographic organization, and it was repeatable for an in vivo aging cohort. We had good agreement to existing track tracing data, and we believe that this better segmentation will lead to improved model development. But something that we're quite excited by is that this insula mapping is an initial use case of the data set, would be to extend this. And one possibility would be to take this sort of unwieldy large data set of histology data and putting it online And so we've been collaborating with Katja Hewer and Roberto Toro using their tool called MicroDraw, which puts these very large histology data sets into a web-based viewer. And so you can see sliding the transparency, being able to look at the aligned myelinated slice over the cell bodies, going back again, looking at labels. And so as we are developing this, there's more work to be done. We hope to be able to do broader levels of annotations. Okay, so I'd like to thank all of my collaborators, in particular Joey and Claude. We've put in a lot of work on this project over many years. And if you have any questions about the details, I'm happy to talk to you. I'll be at poster 2196. And we also have a job posting to extend this project even further. So, thank you.
Thank you very much. There's no question in the app yet, so I happily kick us off. Have you looked at different types of tractography algorithm and how that might change the cortical projection density maps?
So we've been sticking with one particular projection, but we have done other kinds of tuning. So for example, we use a high diffusion weighted imaging, so to essentially try to have more core white matter tracks instead of contributions from extracellular contamination, essentially. And then we also do so anatomically constrained tractography, so looking at the white matter, gray matter interface to try and refine the tracking. But we haven't really experimented too much with other tracking algorithms, but it is a possibility, and the data's there, it could be done.
Thank you. Hiromasa has a question.
Thank you, it's a wonderful talk. I think maybe near insular cortex, I think white matter region is very, very narrow because classroom is just there. So I think this very narrow white matter region can be typically very difficult to tracking. Maybe this kind of narrow area makes inducing some tractography biases. Do you have any just comments about this aspect?
Yeah, so since it is a probabilistic technique, it can effectively get swept away into areas that you are not expecting tracks to go. And so that's why we were doing kind of the tuning that I was talking before, using specialized data, doing the masking. And then, yeah, so that has effectively been able to refine the tracking more so than our initial results were when we were just kind of like, where does it go? So we were definitely seeing spurious fibers going into like visual cortex, for example, at the beginning of this project. But yeah, it is a downside of the technique. It seems to be overly inclusive at times, but we were sort of reassured when going to look at the track tracing data, for example, which is kind of a more ground truth estimation of connectivity, that there was still good agreement in this case.
Okay, thank you.
Sure.
So you are showing data to the tractographic data from anterior insula to the prefrontal cortex.
Yes.
But it's hard to see where it projects, which area it projects, because we have a lot of tracing studies data. So from Joel Price lab, you know that, right? We did a lot of tracing studies. By looking at your picture, I don't know where the projections are. So you have to make some kind of coronal slice where those tracks are. So, but this kind of map, so what is your interpretation, where they project?
Yeah, so we can definitely do a more fine-grained observation of where the tracks are terminating. That's absolutely possible. That's not something that I've prepared for today. And I also noticed you mentioned other track tracing studies are available to include, and that's something that we initially did try to do. So we went to the COCOMAC database and found that for the insula in comparison to other regions, such as in the visual cortex, for example, there was less track tracing data available, and going to the level of the parcellation where basically when you export the COCOMAC data, it's in these large kind of chunky regions, depending on which atlas that you choose. It sort of lost the granularity that is the beauty of the track tracing, which is why I just show one specific example, one study, but there are more studies to compare to. So it could definitely be expanded on further with more granularity, and that's for sure a future direction.
And one more question here.
Sure.
Well, it's a bit of a follow-up on Salim's one. Since you have the post-mortem data and you've got the histology, have you actually mapped, the cortex and identified cyto and / myeloarchitectonically distinct areas to be able to really comprehensively target regions.
Yeah, no, I think that's a fantastic idea, and
I'm-
would really strengthen the analysis for sure. But that's not something we've done. But it is so because of the way the data set is defined. So we have this central space that we can work in. So all of those tracks can be essentially back projected into the histological space and be characterized.
And then you've got the micro draw tool.
Yeah, yeah. So it's possible. Okay, thanks.
Right, wonderful. So next speaker